Good afternoon, everyone, and thank you, JC, for the invitation and the very generous introduction. It's my pleasure today to tell you a little bit about the work that we've been doing at insitro with the goal of using machine learning to bring better medicines to patients. So I think it's not lost on anyone, certainly not this audience, that 2023 was the year when the world suddenly woke up and said, my goodness, look at this thing, AI is here suddenly. Except that it really wasn't all that sudden, and we have actually been living on an exponential curve of technology transformation that has been going on for multiple decades at this point. And the thing about exponential curves is that you don't really notice them so much at the beginning because they look awfully flat, and then all of a sudden you realize that you've been living on an exponential curve. And the important thing about an exponential curve is that it only continues to get faster over time. And that is the thing that we should be keeping in mind as we judge where the technology is today and where it's likely to be in the coming years. So when we think about this exponential curve, it's useful to disentangle its elements because if we understand them, we can potentially replicate them in the life sciences. So this transformation has been driven by four forces. The most visible, of course, is the incredible advancements in AI algorithms, the large language models that we all hear about every day and the capabilities that they keep coming up with. But that rests very much on the vast amounts of data that have been used to train these models. When I started my career in AI and machine learning, which unfortunately I have to say is 3 decades ago at this point, a large data set was one where we had a couple hundred or maybe a couple thousand data points. And now we're living in a world where the number of data points that are being used to train these models are multiple orders of magnitude larger. Of course, the growth in computational power that is used to train those models and the growing base of machine learning talent that is putting all these things together. So when we ask ourselves, can AI help us make better medicines, one of the questions that we need to ask is how can we replicate this set of capabilities, and most importantly, where do we deploy them? Because there's lots of places where they can be deployed and some are more impactful than others. So this is the standard drug discovery and development pipeline, which starts from biology discovery, which is uncovering new biological or therapeutic hypotheses that then feed into drug discovery, making molecules that attempt to implement that therapeutic hypotheses with a molecule, and then feeding into clinical development where we hope to improve the efficiency and success of clinical trials. And this funnel, many of you I'm sure are aware, is not the world's most efficient. I think this is currently the only industry where the success rate of a program is somewhere on the order of 5 to 10%. I used to say that it's the only one except for space travel, but now SpaceX has put that to the side, so I think it's the only one that has this low of a success rate. And when you ask yourself why this is the case, you realize that most programs fail in the clinic because mostly of the early stages of this decision-making process. That is, we are very poor at selecting the right therapeutic hypotheses, the right targets to modulate in the right patient population. So this seems like a very important place to which we would want to deploy this AI technology to try and make this process better. So the good news is that if we want to deploy AI to address this problem, we have the tools to do so. And if you remember my 4 revolutions slide from a couple of slides ago, we have been going through a quiet revolution in biology that JC actually just very nicely articulated in his introductory remarks, where we have a tremendous set of capabilities to generate data at multiple biological scales across multiple modalities. And hence, if you put that together with the AI/ML capabilities, the ability to also derive novel insights. And that is what we hope to leverage in the work to address drug discovery. So how do we aim to do that? So we have built a set of capabilities at insitro that rely on data that is generated both in humans and in the lab. In humans, there is the growing amount of clinical data that is being measured, where our focus specifically has been on high content, unbiased data modalities that are as close as possible to the underlying pathophysiology of disease. So rather than rely on clinician interpretation of what the patient has, which is often very subjective, dichotomized, and very variable, we try and collect as much as we can ground truth measurements from modalities like radiology, pathology, omics in blood and tissue, outcomes such as survival, and then we put that together with genetics, which gives us the important ground truth of cause and effect that is so lacking in the life sciences, where observational data is often very confounded. We supplement the data that we get from humans with data that we generate in our own wet lab, and I'll show you some examples of those capabilities in a moment, where we take cells and similarly, across different cell types, similarly measure them across multiple high-content, unbiased modalities that try and move away from the subjective decision of a biologist on what matters and what doesn't, but really measures imaging, omics, and so on, and similarly puts that together with genetics to understand the cause-effect relationship, in this case in human cells. So that allows us to now understand that causal relationship between a gene and its clinical impact or its biological impact. And there is now, at this point, a well-established understanding, realization that ability to disentangle cause and effect is one of the biggest drivers of success in the clinic compared to anything else that we've done. So this is a very recent paper from 2024. There's other papers that have made that point repeatedly with other data sets that suggest that the probability of success for a drug that goes into the clinic is about 2.6, some will say 3 to 4 times higher for drugs whose targets have human genetic support than for drugs whose targets do not have human genetic support. So this is one of the very few knobs at our disposal today to actually change that dismal statistics that I mentioned earlier of a probability of success of programs going to the clinic of 5 to 10%. So how do we plan to take that forward, and where does AI ML come into this use of human genetics? So as I said, human genetics increases the probability of success by 2 to 3x. But we have a very limited set of human traits for which we have those kinds of measurements that allow us to deploy this type of genetic association analysis. So what we have been able to do, and I'll show examples in a moment, is to use AI/ML to both scale the amount of individuals, human individuals or cellular individuals, as you will see, to both considerably increase the scale and hence the statistical power of the genetic association analysis, and also to identify phenotypes that are sharper, more quantitative, and hence better powered. We also, by virtue of being able to measure and in fact infer multiple different phenotypes, we're able to create multimodal orthogonal support for these different targets, which both increases our ability to uncover new biology as well as to gain confidence around those other clinical significance. So all in all, the sort of hypothesis that underlies what we do at insitro is that we hope to uncover novel high conviction therapeutic hypotheses that are supported by human genetics in multiple clinically relevant traits, with the hope of increasing the probability of success by hopefully far more than the 3x that you get from human genetics alone. So let me talk a little bit about the platform capabilities that underlie this work, and then at the end I'll talk about some of the therapeutic programs that have emerged from these platform capabilities. So this is sort of the block diagram of what we've built, and you can see at the bottom there's the different data modalities that we've put into place: clinical data, cellular data, and also chemical data, which I won't talk about today at all, in order to actually design small molecule therapeutics. All of that feeds into a machine learning and data platform that is kind of at the state of the art, with scientific pipelines, ML-ready data stores, and importantly, 192 H100 GPUs that really drive the machine learning capabilities that we have. And then that feeds into the machine learning driven discovery strategy, which I won't talk about here because you'll see it illustrated in coming slides. So I mentioned earlier that AI/ML can actually close gaps in the data and hugely increase the scale of the data. And what we deploy towards that is a combination of representation learning and generative AI to take data sets that are often underpowered, in the sense that only some modalities were measured for some individuals, but not all modalities for all individuals, and basically use the power of generative AI to fill in the gaps so that you can pretend that you have a much larger cohort with measurements across all of the individuals that are at your disposal so that you can better power the statistical genetic analysis. So this is what that looks like. You might have a cohort where you have some modalities for some individuals and some for others, and then you do a variety of different machine learning algorithms, which I won't get into the specifics, to create a cohort that is much, much richer and better powered than that. So I'll show you some examples of that to make this much more concrete. So first of all, a very cool and actually clinically relevant application, which is the ability to take a DEXA scan and obtain an estimate of liver fat for that individual. Now, DEXA scan, you talk to people, they'll say it can measure bones, maybe muscles, but no one thought it could actually measure liver fat. And it turns out that you could actually impute liver fat with very high accuracy, and you can see that when the machine learning is imputing liver fat, it's actually looking at the liver, so it's actually doing the right thing. And what you're able to do now is you can get, as you can see in the graph on the top, on the left-hand side, an imputation of liver fat that is quantitative that has an R-squared of 0.63. Now, the other approach for measuring liver fat is you put someone in an MRI machine. It costs several $1,000s. You can get a DEXA scan at your local gym for $50. So that is something that has clinical diagnostic relevance, but also, as we'll see, it powers the discovery work that we're doing at insitro. Now, it turns out that DEXA scans are not the only modality from which you can impute liver fat quantitatively. You can impute liver fat from circulating metabolites that you can get from a test tube of blood, or from circulating biomarkers that you can similarly get from that test tube of blood. The R-squared is not quite as high, but it's good enough, as you will see, for the kind of work that we're trying to do. Another example of imputation comes up in the context of oncology, and JC talked very nicely about the power of the TCGA cohort and the amount of data that has been collected there. We leverage that cohort as well as others in order to see how much information one can extract from a standard biopsy sample. Now, the thing about biopsy samples, and specifically the H&E staining of a biopsy sample and viewing it under the microscope, is that it is probably the data modality that is most abundant in the context of clinical care, because everyone who comes in with even a suspected case of cancer, as well as multiple other diseases, IBD, Crohn's, and others, gets an H&E biopsy sample. And so it's very, it's routinely collected, increasingly in digital form. It clearly has high content at multiple biological scales that range from, coarse organ grain structure like vasculature all the way down to what happens within a single cell. But you give this to a human pathologist and you get 3 ordinal numbers as the output. And so a ton of information is left sitting on the table. So we basically took those H&E biopsy samples, learned a foundation model of histology. Others have done that subsequently to our work or in parallel to our work, and are able to impute multiple clinically relevant biomarkers from that H&E alone, things that typically would similarly require an additional test, often costly, often time-consuming, cancer patients requiring, needing to wait longer to get the results. It turns out that you can get very high accuracy results of DNA somatic alterations, as well as RNA-defined subtypes that have significant clinical consequences in terms of the success of different treatments. Moving to the in vitro side of the world, a technology that we've developed that we call insitro paint enables us to virtually stain live samples. So rather than fixing a sample and then staining it with different fluorescent dyes in order to measure different cellular markers, you can actually take an unstained sample and get results that are effectively indistinguishable from the stained version, but you don't have to fix and stain the microscopy sample in order to do that, which means you can track those cells over time as if they were stained. And so this is, I don't know if I can, how do I control the mouse from this? Thank you. So here you can see, for example, thank you so much, the unstained sample on the right. If we can click on that again, that would be awesome. You can see the unstained sample on the right, and here this is a stain of lipids in those. These are hepatic liver cells, and you can see that there's, you can basically stain that and see the lipids moving around and being accumulated. And then you can, on the right side, you can see that you can now, for example, over time, test the effect of a therapeutic intervention. So you can see that over time, as you introduce a drug into those hepatocytes, the unhealthy cells turn into more healthy cells by having less lipid accumulating in those samples. So this is something that you could actually now test therapeutics live in action using this approach. So if we move to the next slide, I guess I could do that. No. Okay. Great. So I showed you this example in liver fat. Now I'm going to show you one example in the other therapeutic area that we're very active in that will come up in a few minutes when I talk about the actual therapeutic programs, which is looking at this point at motor neurons that capture the biology of an ALS patient, Lou Gehrig's disease. Now, many of you are familiar with ALS. ALS is a devastating disease. It strikes people at any age, sometimes as early as their 30s or 40s. And basically, it's a death sentence within 3 to 5 years. So very few people survive past 5 years. We have a therapeutic program in ALS and have developed capabilities to generate, measure, and perturb and measure motor neurons. And what you see here is the ability to take motor neurons that have only cell morphology and impute from those images of motor neurons multiple ALS-relevant markers of disease. So on the left side, you see something that takes basically just, again, neurons that all you have is their morphology and are able to impute the mislocalization of a critical disease protein, TDP-43, that's the hallmark, almost the definition of ALS. You can just by looking at the cell predict whether TDP-43, which is not measured, is mislocalized from the nucleus to the cytoplasm of the cell, which allows us to basically assess whether these are disease cells or not simply by looking at their morphology. On the right-hand side, perhaps even more surprising, is the ability to impute the gene expression, the transcriptional profile of multiple ALS-relevant genes just by looking at cell morphology. So you can look at the cell and assess whether genes that are ALS-relevant are overexpressed or underexpressed relative to wild-type healthy cells. All of that allows us to perform screens in a much more effective way to identify, as you will see, disease-relevant reversion of those phenotypes. So, coming then to where all this is useful, so phenotypic imputation is all great, but then how does it give you drug targets that you can now potentially use as therapeutic interventions? So what we have shown is that we start by generating and aggregating high-content data. We impute these additional disease phenotypes and then learn disease axes in the space that in this high content space. So what you see here is a movie that shows you cells that morph, a disease axis that shows the movement from a healthy to a fatty liver. The disease pushes the cells from the healthy side to the unhealthy side. Our goal is to find things that go in the other direction, and we'll show you that example in a moment. This is similarly a disease axis for ALS. So what you see here is these are synthetic images, by the way. It's a visualization of the disease axis that was uncovered by the machine learning. You can see a movement that takes a healthy neuron, which is what we started out with, and turns it into this unhealthy neuron. And you can see that the soma elongates. There is a reduction in the axonal arborization. And so you can see that the machine discovered in this high content image this disease axis. And then subsequently, those disease axes that leverage these machine learned traits are allowing us to do genetic discovery, both in experiments of nature and experiments in the lab. So what I'll show you now is that this all really works. So this is not just fancy machine learning, but actually has given rise to therapeutic programs. And I will show an illustration in 2 of our programs that are furthest ahead. The first is MASLD. I'll also briefly mention some early work in obesity. So MASLD is fatty liver disease, and I'll show you some of the work that we've done there. This is our furthest ahead program in the sense that it's going to, that we're close to pushing that into the clinic in early 2026. The next, and then I'll show, the next program is the work that we've done in ALS in partnership with our collaborators at Bristol Myers Squibb, and I'll show you the targets that machine learning has uncovered in that indication as well. So starting with our work in metabolic disease, I mentioned earlier our ability to impute liver fat from multiple different modalities. So why is that important? It's important because we want to uncover genetic modifiers, genetic modulators of quantitative liver fat in humans. So what you see here is a Manhattan plot, and some of you I'm sure are familiar with this, but for those of you who aren't, the x-axis in the Manhattan plot is your chromosomes organized linearly across the x-axis. The y-axis is the negative log p-value or statistical significance of an association between different regions in that genome and the trait in question, which in this case is liver fat as ascertained from MRI. So as I mentioned, MRI is a complex, expensive modality. Not many patients have had an MRI and also have genetics, genome sequencing done. And so when you look at this Manhattan plot, by the way, it's called Manhattan plot because it's supposed to be evocative of the Manhattan skyline. So you have multiple towers indicating these statistically significant genetic associations. This doesn't really resemble Manhattan, more like maybe the cornfields of Iowa. So not a great starting point for a lot of new targets in drug discovery. There's only 7 loci that came out of that. As we move towards these better powered modalities where we have additional humans, where we have measured metabolites, DEXA metabolites or biomarkers, all of a sudden you have a much larger N, which allows statistical significance to emerge above the noise. So we move from 7 loci to 15 to 55 to 314 loci in the biomarker modality that have suggest that where there is a strong association that passes so-called genome-wide significance with the liver fat trait. Several things came out of this, several known targets that previously had not had human genetic support, but that are known to be implicated in the disease, came out for the very first time. So FGF21, for example, is one of the most successful clinical targets that's moving forward in clinical development towards liver fat, towards fatty liver disease. Never had human genetic support. First time that it came out is in that graph at the bottom. As well as, fortunately for us, not just known targets came out, but a whole slew of novel targets. Now, the novel targets are actually kind of interesting because not only is each of them interesting in its own right, they also elucidate pathways. So what you see here, for example, is the previous genetics for MASLD, the name of the disease, overlaid on a pathway diagram that corresponds to the different processes that drive accumulation of fat in the liver. So there's the uptake of fat at the top, there's lipid droplet regulation in the middle, there is de novo lipogenesis on the left-hand side. This is the process by which things that are not fat, but rather sugars, are taken up into the liver and form fat in the liver, de novo lipogenesis, and then fatty acid oxidation and so on. When you look at this diagram and you ask yourself which process actually drives the disease, the answer is, I don't know, none of them seem to really bubble up. But when you take the genetics that came out of our much better powered analysis and you overlay that on top of the same pathway diagram, de novo lipogenesis comes out as the strongest by far pathway that seems to drive the disease. When these results were presented at the liver meeting, all the hepatologists in the room and the clinicians were like, yep, yeah, we kind of knew that, because the thing that drives fatty liver is all of the sugars, the carbohydrates that we consume in our diets, the fructose and the glucose, but there has never been the genetic support to really support that hypothesis, so this is the first time that that came out. That drove the selection of our therapeutic targets, focusing on the de novo lipogenesis, and basically also modified the way in which we do our lab experiments so that instead of subjecting the cells that, in the lab to free fatty acids, which had been the typical discovery approach used in the discipline, which focused on the uptake of fat into the liver as the mechanism, we put in the substrates of de novo lipogenesis, glucose and fructose, and you can see that you get accumulation of fat in the liver on the left. These are these green little droplets in the cells. Our target, which is our furthest ahead target in the middle, significantly ameliorates that phenotype, as does DGAT2, which is a target that is currently in the clinic that we're using as a positive control. And what you see here is that our target modifies this phenotype as well or arguably better than DGAT2. So we didn't just come up with one target, we came up with 5 targets that we've pushed ahead as quickly as possible. There's others behind them in line. And you can see that of the 5 targets that we moved, in this case, into an animal model called the diet-induced obesity mouse. It's one of the more translatable animal models out there. 4 out of the 5 considerably decreased the accumulation of triglycerides in the liver. We were actually happy that it was 4 out of 5, not 5 out of 5, which would have felt kind of freaky. So this was a very pleasing result from our perspective. This is the furthest ahead target again, target A, and you can see this is a much more translatable phenotype. This is actually a version of the phenotype that is used for regulatory approval by regulatory agencies, which is this steatosis phenotype in livers. So at the top you see a nice healthy red liver on the right. This is what your liver should look like. On the left is your liver on fat, which is, all these white droplets and the pinkish texture that is lipid accumulation in the liver. And what you see on the bottom is basically, again, the fatty liver on the left and the modulation of that steatosis phenotype in target A and DGAT2. And you can see that in this experiment, which is a very relevant one, our target actually considerably outperforms DGAT2. So fortunately, we were able to craft a very nice partnership with Eli Lilly, clearly a very strong leader in the metabolism space, on 3 of our lead targets, target A as well as 2 others, to address the drug discovery efforts. We have small molecule capabilities in-house, but in this case, we're using other therapeutic modalities, siRNA and antibodies, and they're very helpful in helping us make molecules that will actually be efficacious towards those targets, and they're helping us do it way faster than we could have done it, for example, with a CRO and with very high quality molecules. So we're very privileged to have had this partnership with Lilly. And so summarizing the MASLD case study, we identified multiple. I showed you 5. There's others behind them in line. Novel genetic modulators with strong multimodal support across multiple different modalities in human genetics. Of the 5 that we tested in animals so far, 4 have significantly reverted liver fat, and there is proprietary compounds that are moving towards the clinic with IND-enabling studies scheduled to begin in next year, early next year, and hopefully an IND expected in early 2026. And importantly, from the time that our therapeutic area head said we're going to go after MASLD to the time that we had the first proof of concept in the animal model was 18 months, which is a very rapid discovery timeline. And so now we're looking to replicate that using our platform. This is very early results on obesity, and specifically we're focusing not on central obesity, which is where the incretins, GLP-1s are so successful, but rather on changes in where adipose is actually deposited. So not how much appetite you have, but is adipose deposited in healthier regions versus unhealthy. So there's actually now fairly well understood that fat that accumulates around your thighs and buttocks is actually somewhat healthy. If it accumulates in your abdomen, very bad. And so can we actually modify that phenotype? It's never been quantitated before, and we can do that using our machine learning techniques. And so this is an early GWAS of that. So very briefly on cellular machine learning, the work that we've done in ALS, this is a much shorter segment, but I think it really highlights the sort of diversity and flexibility of the platform that we've built. So here, the focus has been on our in vitro capabilities because it's very difficult to get human clinical data in a sufficient number of ALS patients, given that it's a relatively rare disease. So we've created a library of so-called induced pluripotent stem cell lines that capture the diversity of the ALS genetics in cellular systems. We showed that we can differentiate those IPS lines into motor neurons actually in a record-breaking 10 days, and that those lines exhibit the pathophysiology of ALS, specifically that TDP-43 mislocalization that I showed you earlier, as well as other more functional phenotypes that are downstream of that. We're able to phenotype those lines using capabilities that we've built using imaging and transcriptomics, and I already showed you the really cool disease axis in a previous slide that allows us to gain conviction that the patients, that the lines that exhibit the disease genetics are truly different to the lines that are healthy, allowing us to then screen for modifiers that revert that phenotype. So the reversion leverages a platform that I didn't talk about before, but it's a fairly unique capability to our work at insitro. It's called POSH, or Pooled Optical Screening in Human Cells. It is a pooled CRISPR screening platform that takes a pool of cells, a pool of CRISPR guides, introduces the CRISPR guides into the cells so that each cell on average gets about one guide. So now you have a pool altogether of cells that are genetically diverse. You can now phenotype those cells using high content imaging or transcriptomics, but I'm showing you imaging across multiple different modalities. You can look at the morphology of the cells. You can look at the localization of proteins such as TDP-43. You can look at the live cell dynamics that I showed you earlier. And having done that, you fix the cells and then you sequence the barcode that tells you which cell got which guide. So you can think of it as a sequencer inside a microscope, which gives you the ability to, in a single pool of cells in a single plate, to do a full-blown CRISPR screen for reversion of these high-content disease-relevant phenotypes. And we can now, we've done genome-wide CRISPR screens, and we can go from a disease model to these modifier hits in about 3 weeks. So this is where the project currently stands. We have done this at-scale POSH and perturbation screen on this disease axis. We have identified a set of hits, and we have taken those hits and done additional what we call credentialing work, and convinced ourselves that those hits in fact revert multiple functional consequences of ALS in those cells. We've also convinced ourselves that they don't kill the neurons, which is also important. So we are currently taking those targets and prosecuting them in our drug discovery pipeline, which I haven't talked about, but we're very excited about those because they seem to be upstream of where a lot of the other drug programs currently are working on, but needless to say, there is no current drug against this disease that is actually effective. So I was asked as part of this presentation to talk not only about the science but also about the culture. So I will end this presentation with a little bit of that, which is how do you build a company that is truly at the intersection of technology and biology? Because those two disciplines are ones that don't often talk to each other and have a very difficult time communicating, not only because the language is different, but timelines are different. Engineers are used to working on 2-week sprints. Scientists, sometimes you ask them for annual goals and they say that's too fast for the science to progress. And they think about the world in very different ways. So engineers love patterns, as simple as possible. You know, if it's a straight line, that's awesome. Like a linear regression explains a lot of the data, that's as good as it can get. Scientists often look for the exception, the outliers, the things that don't fit the pattern, because those are often the kind of the end of the thread that if you pull at, gives novel biological insights that no one has looked at before. So these are very different ways of thinking. So how do you take these two groups and put them together in a company where they're actually working together. So first of all, it starts from the top. And we've been very fortunate to recruit a group of leaders that come from very diverse experiences with people who have, like our CSO, Philip Tagari, 45 years of drug discovery experience. Mike Rothenberg, our chief medical officer, has put multiple drugs into the clinic successfully. But also people from our, technology background. So Emily Fox, who came to us directly from Stanford, where she was a professor there, also built a health AI group at Apple. So you take those people, and if they collaborate at the top, that definitely trickles down all through the organization. You create a culture where people work together in cross-functional teams. So while at insitro, people report functionally. So if you're a machine learning scientist, you report to Emily Fox. And if you're a drug discovery person, you report to Philip Tagari. But all of the actual work happens in cross-functional teams where people collaborate. Not at the end. It's not that at the very end, after the experiment is done and it's time to analyze the data, they come together. They collaborate from the very beginning. From the time, not even that they design the experiment, but they define the problem. And we find that when you put these people in the room together at day one, then they not only come up with better answers, they come up with better questions. And this is all enabled by what is a very unique build for a biotech company. So you can see here the sort of mix of people, the composition of the company at different points in time. And so the gray is basically G&A, but you can see that the ratio on the R&D side between lab and compute is about 35-65. And if you actually put the automation people on the compute side, which is probably appropriate, then it's more like 40-60. And that is what enables this deep equal partnership across these disciplinary boundaries. And without this, insitro would not be where it is today, because a lot of the ideas that I presented to you came out of this cross-functional collaboration at the very beginning. So this is actually my last slide, which is, we've heard a lot about multiple applications of AI, and everybody reads about AI agents and chatbots and things like that. But AI can also serve what we believe is a truly aspiration goals, which is our mission statement, to bring better drugs faster to the patients who can benefit most through machine learning and data at scale, or as we like to boil it up, building a pipeline through a world-class platform. So thank you very much.
We have time for one question. If I look at the 1990s or the first 10 years, the number of parameters that we look at in a drug discovery program was 10, 20, and never mind genetics. So would you show us DEXA scans before we heard about the possibility to interpret important data from EKGs from light microscopy. So do you think that with multi-parametric, apart from genetics, multi-parametric assessment of compounds and targets, the chances are going to be 3x, no, 30x? So what do you think?
Well, I think that 30x is a lot given 5% times 30 is more than 100, but yes. I think that the data on support from human genetics is very, very strong, and I believe that if you have support across multiple modalities, each of which corresponds to a disease-relevant trait, you certainly gain a lot of conviction. And actually one of the slides that I have that I didn't show is a demonstration of that, where we actually took the targets that in our work in MASLD received multimodal support from DEXA and biomarkers and metabolites, and we asked how many of them, if you go back to the ground truth, limited as it is, which is clinical diagnoses of MASLD, how many of those replicate there? The ones that receive multimodal support are more likely to replicate there. So admittedly, it's a crude metric of success because we know that is an underpowered cohort in itself, so it's probably missing some stuff. But it certainly gave us confidence in that multimodal support, and especially if you also add some of the cellular confirmation as well, is going to increase our probability of success, hopefully far beyond the 5 to 8% that it currently is.
Excellent. Well, thank you so much. Let's Thank you.